Hi, today's video is about carbohydrates and sugars part 2, polysaccharides. This is for the exam board AQA for A-level biology. So we need to know how polysaccharides are polymers, and polymers are many monomers or simple units which are bonded together in a chain to form larger molecules. Polysaccharides is when there's a condensation of many monosaccharides or disaccharides to form these polysaccharides, such as starch, cellulose, or glycogen. These are the three examples we need to know for our A-level spec. Starch is the plant's store of glucose. It is a respiratory substrate. Cellulose is used in plants' cell wall for structural support. And glycogen is the animals' store for glucose, or the mammals' store for glucose. And it's also a respiratory substrate. So zooming into starch, we have to establish what starch is. Starch is basically two polymers. It consists of two polymers of alpha glucose, and you need to know the names of these two. One is amylose and the other is amylopectin. So starch is found in the starch grains in plants. Starch is insoluble, therefore yep, it's an insoluble store of glucose, so later on you find out how it doesn't affect the water potential of the plant. So insoluble, we find out how amylose and amylopectin are both insoluble. Zooming into the structure of amylose, it's 1 to 4 glycosidic bonds. So carbon 1 and carbon 4 will partake in these glycosidic bonds. The amylose structure itself, it coils into a helical shape, and you can describe them as straight spirals. And these coils that form, as you can see here, they can compact together almost like a spring, and this compactness can fit a lot of glucoses in less space, as you can imagine. I've drawn these white dots to show, just not scientifically, however, these white dots could show glucose, and a lot of these glucoses form. You compact this together, a lot of glucoses in less space. So now, the fact that it's insoluble, as I said before, it doesn't affect the water potential. This is the sign for water potential. And the fact that it doesn't affect water potential, it means that therefore it won't affect osmosis, or it won't lead to damage of the cell or organelles, and therefore it won't lead to osmotic lysis, which is the bursting of the cell due to osmotic pressure. Next, we move on to amylopectin, which is the other type, or the other part of starch, another polymer of starch, if I should say so. So amylopectin contains both 1 to 4 glycosidic bonds and also 1 to 6. These 1 to 4 glycosidic bonds, they form these straight chains like this. However, these 1 to 6 glycosidic bonds lead to branching out of molecules. Therefore, we learn that this structure is a branched structure. As I said, 1 to 4 gives a chain structure, 1 to 6 gives a branched structure. As we know, it's 1 to 4 carbons taking part and 1 to 6 carbons taking part here. Okay, so the fact that amylopectin is branched, as you can see in this just simple drawing here, it means that it has an increased surface area. There's more surface that is exposed. So the fact that there's an increased surface area, we can say that there's an increased surface area for rapid hydrolysis back to glucose, because this molecule, it can be hydrolyzed to form glucose once again, because we know how starch is a store of glucose. Okay, also, as I said before, because it's insoluble, it won't affect the water potential, therefore it won't affect osmosis, therefore it won't cause damage. Now for the starch, I have got a nice question, which is explain one way in which starch molecules are adapted for their function in the plant cells. So each of these three are worth 2 marks each, and the one thing you can say is it's insoluble. Insoluble, therefore it won't affect the water potential of the plant, so it won't damage the cell via osmosis. Another point you can make is helical, therefore it can compact to store more glucose in less space. And another thing is the fact that it's large means that it won't leave or escape the cell. Okay, now we move on to cellulose. Cellulose is formed from beta glucose. Compared to starch, which is formed from alpha glucose molecules, cellulose is formed from beta glucose. So we see how in this image, which can be found on some past paper questions, this image shows that the position of OH and H groups in the C1 is opposite to that of starch. So in starch, we see how alpha glucose is used. Cellulose, we see how beta glucose is used. They're both isomers, beta glucose or alpha glucose. So therefore, there will be some differences, structural differences. Okay, next thing is cellulose can be found in plant cell walls to provide structural strength, and they are basically long straight chains that are lined up parallel. So this is just one chain that you can see here, and a lot of these chains line up parallel as seen here. So when they line up parallel, what happens is each of these chains, they will form hydrogen bonds with each other. Individually, hydrogen bonds aren't strong. However, lots and lots of these can collectively be very, very strong. And when these structures form as seen here, they form a microfibril or fibril structure. The fact that it's insoluble means that it won't affect the water potential of plant cells, therefore, it won't lead to any potential damage via osmosis. Also, this provides strength to plant cell walls. And now I've put a question once again, a potential question which could come up. Explain how cellulose molecules are adapted for their function in plant cells. So one point you could say is formation of long straight chains, as I said here. And long straight chains, they do form glycosidic bonds via condensation reactions, just extra knowledge. So formation of long straight chains, second point being, they line up parallel and are held together via hydrogen bonds to form fibril structure. So as I show here, the hydrogen bonds. And this forms a fibril structure, and then it also, therefore, it provides strength to the plant cell wall and prevents damage. This is a 3 marker, so you need 3 clean points. Okay, next we move on to glycogen, which is the third polysaccharide we need to know. So glycogen is very highly branched. One thing you need to know is that it is more branched than amylopectin, which you could potentially be asked to compare this to amylopectin, for example. Glycogen once again is made up of 1 to 4 and 1 to 6 glycosidic bonds. As you might have guessed, 1 to 4 glycosidic bonds form these straight chains or helical chains, and these 1 to 6, they form the more branched structures. So because the reason why it is more branched than amylopectin is there's a higher ratio of 1 to 6 bonds. So you can imagine there to be lots of these. So glycogen is found in the liver and muscles, and as I said before, it's very highly branched, therefore an increased surface area to store more glucose and for a more rapid hydrolysis back to alpha glucose. So you can imagine if there is a higher surface area, rate of reaction will have increased and the formation of glucose will be quicker. And once again, like the other two polysaccharides, glycogen is insoluble. The fact that it is insoluble means that it won't affect the water potential of the cell and it won't form osmotic lysis as I explained before. Just some extra knowledge which I think you will need is the extra knowledge about glycogenolysis, which is the hydrolysis of glycogen back to glucose for when animals or mammals need glucose more, because our blood glucose levels, as some of you might know or have studied diabetes, for example, blood glucose levels can fluctuate or change. So our body needs glycogenolysis if our blood glucose levels go too low. So the hydrolysis of these glycogen molecules can help to counteract this change. so it can counteract the too low blood glucose levels. Some more extra knowledge is that this glycogenolysis happens in muscles and livers mostly. That's all for polysaccharides, and I will be moving on to fats in the next video. One thing is testing for these biological molecules. I will make a separate video where I test for all of the different biological molecules you need to know for topic 1. Thank you for watching.